Hello and welcome. I'm at the Atheist Convention in Philadelphia now, so this week's video is a small portion of the conversation that I had about a month ago with Dr. Zach Hancock, an evolutionary biologist who specializes in population genetics and phylogenetics of beach voles. Today, he responds to the creationist talking point that evolution cannot create new information. Take it away, Zach. They continually make the claim that evolution is incapable of creating new information.
Mm.
That yes, there's genetic mutation, but it's always just a repeat or a rearrange of the information that is already there. It's never new information.
Mhm.
How do you handle that question?
Yeah, so there's several different ways you can approach this. I've seen people try to pin them down on what information is.
Mhm.
That's not really a route I go. I just try to assume that by information they mean something like a phenotype, like a genotype that codes for a phenotype. I assume that's what they mean. I try to give them the benefit of the doubt and not try to squash them on the definition of information. So if we assume that by information we mean generate something new, something novel, either a novel protein or a new biochemical pathway, something like that, right? We take that as being information. Then it's very easy to refute this because we have so many examples of this exact thing happening. And we even know many different ways in which it can happen. So at the very basic level, we have what's called de novo gene synthesis. And this is where you have a non-coding region of DNA. That's a region that doesn't make anything, that then through subsequent mutations acquires what we call an open reading frame. That's where you have a start codon and then enough bases down and a stop codon. And this basically is what you need to tell, the ribosome start coding at this end and stop coding at this, and then it will generate an amino acid string, right? And this can happen completely randomly in non-coding sequence. And it's actually really easy to pick these up. So what we can do is we can take two closely related species. This is famously recently done in yeast. Yeast is perfect for this because we have baker's yeast that's sort of like the domesticated yeast. We really understand their genome super well. And then there are wild yeast, right? There are like other species of yeast as well. And so we can go out into nature, we can take these wild yeast and say, okay, what genes do they have that we don't see in baker's yeast? And since we know that they are very closely related yeast, that any genes that the wild ones have must be novel. They must be some new gene. And so what we have found is that there are populations of wild yeast in which these brand new de novo genes are polymorphic. That means some individuals have them and some individuals don't. And this tells us that they must have emerged and they're spreading through the population but haven't become fixed yet, right? So we have sort of an empirical example of novel genes emerging. And they have really cool characteristics. So very often de novo genes are very short. They're often pretty simple because they haven't had time to become elaborate. They usually have a little bit lower transcriptional efficiency, which means that they're not transcribed like normal genes quite as efficiently yet because these things take time to build up. But we see things like selection acting on them. We see them behaving in functional ways with other protein interactions. So we know that they're doing things for the organism, and we know they're spreading through the population. So that's sort of a de novo from non-coding gene. This is a bit of a rarer sort of way of getting new information, but it's literally new information from things that didn't have information previously. A bit of the more standard way is by gene duplication. So you basically have some existing information, you duplicate that information either in tandem or by recombination to some other place. You have now two copies of it. And since you now have another copy, it's freed to explore mutational space, right? Because selection is keeping that single copy constrained, right? Because if it mutates and breaks, then you can't do anything with it anymore. But if you have two copies of it, now one of those copies can become is more mutationally permissible, right? And so there's a couple of different fates that this new copy can take. The new copy can either undergo what we call subfunctionalization, and this is a really interesting way of generating complexity in genomes. So if it undergoes subfunctionalization, what that means is since you have two copies, some mutations can occur in one and in the original one, such that they're both degraded in some way that they now rely on each other. And this actually creates what's called epistasis, so interactions between genes, and they're both necessary to be there. And we believe that a lot of the complexity in eukaryotic genomes were actually generated by this process, that you have lots of duplications, they each kind of mutate in their own way, and now they're all linked in this complex web of interactions because of this subfunctionalization activity. So that's one way. But another way that they can actually that they can gain new information is what we call neo-functionalization. So the
Mhm.
original copy stays normal, keeps its original function, and the duplicate now acquires a new function. Right, so it gets a mutation that now interacts with proteins in novel ways. It has different binding affinities, etc. We have lots of great examples of these as well. The best studied are in Drosophila, because that's a fruit fly that we've studied in labs for over 100 years at this point. And so we very well know what their genomes are like. So we can take sister species and we can say, okay, these are duplicates in each of these species that are unique to these species. Do they seem to have novel functions? Do they seem to have novel binding affinities relative to the parental copies? And we have hundreds of examples of this. So we know neofunctionalization happens, we know subfunctionalization happens, and that's a really simple way of increasing the information content in the cell.
How do you learn that? And what I mean by that question is, how do you learn what function a particular gene has?
So this is there's these kind of molecular assays in which you can take some protein structure because you know what it codes for, and you can synthesize this protein. Right. So if you know the amino acid, you can synthesize it in a lab and then you can subject it to different substrates. So you can say, okay, does it bind to this substrate? If so, what's the affinity? How well does it bind to this substrate? You can
Oh,
also find out function by knocking it out. So if you
uh-huh.
have this neo-functionalized region, you can just like snip it out of the genome and then say, okay, how does development happen? Does anything change? Do we see a pathway fall apart, right? And that will tell you, like directly, okay, well, this was serving an important enzymatic, process within this pathway, and so we know that by shutting it off, it must have been, like we've now broken it, right? So by either breaking it or by actually subjecting it to a molecular assay, we can determine what these proteins are doing.
That is so cool.
It's pretty neat, yeah. A third way of generating new information is by what we call retrogenes. And
Okay.
retrogenes are really cool. So if you take a protein, an mRNA comes and they transcribe it. And so you get this mRNA transcript. Well, that mRNA transcript is currently what we call pre-processed, which means it still has introns. So in eukaryotes, their protein coding regions have exons and introns, and the introns have to be cut out, and then all the exons are snipped together. Right? So actually, your genes are bloated with things that have to get cut out. And so that happens after the initial mRNA transcript is formed. Now, once you get a post-processed mRNA, where you've cut out all those introns, sometimes those can accidentally get reinserted back into the genome by reverse transcription. And so you have the
Mm.
original copy in one place, and then it gets, you make the transcript, it's supposed to go out and be turned into a protein, but it gets picked up by what we call a reverse transcriptase. It's just an enzyme that will force it back into the genome. And it just like adds sort of like a duplicate copy. Most of these become what we call pseudogenes. So they degrade, they don't do anything. A lot of the junk in your genome are these sorts of things, they're these retro elements, right? But every once in a while, they will actually acquire a secondary function. And we can tell that they've acquired a function because they're transcribed at really high levels and because they're sequence constrained. And that means that when a mutation happens there, it's deleterious. And so selection gets rid of it, right? So it's kind of maintaining that sequence identity. So those are sort of the three big ways in which we understand how new information gets added to the genome. And again, I can't stress this enough. We have hundreds of studies showing these processes in labs and in nature. This is an incredibly well documented process that when creationists say mutations don't add new information, I'm just baffled. It's like looking at the sky and saying it's green. I don't know how they can do it. But yeah, basically the takeaway is you can either get new genes from regions of the genome that didn't make genes originally. You can get them by just duplicating your gene and then having one of them turn into something new, or you can get them from a transcript of one gene gets reinserted someplace else and then gets co-opted. That's the basic takeaway.
If you are a creationist, I would love to hear your response. Live your life.